Swayam Prabha. Digital India. Educated India.
Hello all, welcome back to our sessions on precision oncology. In the last session we have seen on the role of cell culture in precision oncology. These following two sessions will be an extension of the last session where we will be working on more of preclinical models for cancer. So, such as what are the disadvantages of cell culture we have clearly discussed. So, we have discussed why the generations passaging of many cells to generations may be losing the cells, the tumor cells, the cancer cell lines may be losing their original phenotypes or their genotypes, and the original characteristics of the tumor will be lost. So we have really come into terms why is it important to have a proper two-dimensional or a three-dimensional current color cell line which is representative of the original tumor for each specific patient. So further moving on, we will be discussing about the preclinical models here in these two sessions. For the first half of this session, we'll be focusing on the patient-derived explants followed by organ-on-chips or the tumor or the organoids models, and then in the next session we will be discussing in detail about the patient-derived xenografts or the mouse models used for cancer. So why are we using so many preclinical models? Why is it necessary? Because it is like each tumor is it's different for each individual based on the genotype, and some of these tumors are very heterogeneous. So it may not be enough for a single population of cells such as a cell line to represent the complete genome or the complete tumor characteristics. So moving on, here is the introduction of the first slide. So this is a model of a tumor microenvironment. So if you recollect well, we have been discussing what is the role of an immunotherapy during our immunotherapy sessions. So how the immunotherapeutics or the ICBs, immune chain blockades, they have to be very efficient in moving the tumor from an immunosuppressive cold tumor microenvironment to a pro-inflammatory hot tumor microenvironment. So here in that regard, we have discussed in detail about the PD1 with the PDL1 blockers. Now here, if we can clearly recollect, there are many types of cells, it's not a single homogeneous cells. So this shows a simplified schematic of the tumor microenvironment. It is very organized landscape, and it includes the non-malignant stroma such as your the myeloid immune subcells, the cancer associated fibroblasts, the blood and the lymphatic vessels. So generally with this tumors, with this immunosuppressive immune filtrate, they resemble the chronic inflammation, and they are termed as immune cold and are associated with poor prognosis. Such immune infiltrations, you have the M2 macrophages, the myeloid derived suppressor cells, that is your MDSCs, then you have the CD4 plus T helpers and the regulatory Treg cells and the terminally exhausted CD8 T cells, the regulatory B cells, the CCR21 cells, all the monocytes, and these are which and this whole and the mature dendritic cells which are enriched in the immunoregulatory molecules. So here you can clearly see the immune filtration which resembles an acute inflammation, and it exhibits a pro-inflammatory or a hot immune infiltrating which includes your elevated levels of the CD8 T helper, CD4 tells the natural killer cells, and the group 2 innate lymphoid cells. The M1 type group 2, the M1 type macrophages, eosinophils, CX3C chemokine receptor 1 monocytes. So these tumors are generally excellent candidates for immunotherapy strategies. So this so taking into regard the vast immune landscape of tumors, which spans from an immune cold to an immune hot, and which is closely linked to the response of either an immunotherapeutic or a targeted therapeutic. So this there are to make a better permeable targeted drugs, it is very important to prepare a preclinical model which represents the original tumor cell. Here we have taken only example with respect to an immunotherapy, but any kind of a tumor, a heterogeneous tumor, for example a lung cancer, the EGFR, the wild type, and the EGFR mutant, which has a targeted therapy for such as your gefitinib, the tyrosine kinase inhibitors, they all have to be well characterized for each particular individual. So this pharmacological spectrum it should include a spectrum of tumor directed or immune directed drugs or antibodies that would enhance the immune mediated tumor cell killing. It is very important to have very good preclinical models which are representative, or which we call them as avatars of the same patient tumor. So, if you mimic these tumors, or if you mimic this tumor microenvironment in an in vitro condition, in vitro, that is in a lab condition, yes, can we see and give the same drug as the patient is being administered in the clinic and look for the response of this particular tumors to the drugs? Yes, can we predict the outcome of the therapy in a preclinical model or in the laboratory? Yes, many studies and several much, the whole pharma is going in this direction. So the different models which are used here are the spheroid, the organoids, or the organ on chips, which are very well characterized, the patient derived xenografts, your mouse models, and the patient derived explants. So spheroids are nothing but it could be even a single line, a cell line, which is allowed to grow in the presence of in the presence or absence of drug, and that too the cells are allowed to form clusters rather than forming a 2D monolayers. So as a result, we would be able to have a necrotic zone of necrosis along with a line of living cells. Yes, we can produce some hypoxia like conditions by growing spheroids. Organ on chips, several researchers, and it's a lot of engineering technology getting into it to develop many organ on chips for many of the cancer models such as the breast, lung, and very well colon and the pancreatic model, which we'll be seeing in this session. Coming to the patient derived xenografts, very well explored. So many labs have come up with mouse models, several mouse models such as and based on the immune status, the nude mice, the skid mice, the NOD skid mice, several mice models they have come into play, which we will be talking in the next session. And then the explant session, patient divided explants, which are ex vivo cultures where the surgically resected tumor is grown in the lab for a couple of days, and the same drugs, or maybe to try the efficacy of the novel therapeutics on this tumors, can be tested. So this patient-derived explants have been very well used to develop novel therapeutics, and it's primarily very important to immediately see the effect of a therapeutic drug on the patient, on the same tumor to which the same patient will be taking up the same drug. So before the outcome of the drug, or the patient how the drug before the patient himself gives an outcome, or how to see the progression free survival and all that, this particular treatment can be seen in the patient derived explants. So, and the drug response can be studied, and we can in all this particular or preclinical models, the endpoint analysis is to study the response of the drug, is to study for any other several variant DNA, RNA, or the protein changes. For all the studies here, it's to check for the drug response, to check if the patient if this particular tumor can respond to the drug, to the same drug to which the patient is being given therapy, and at the same time can we predict the drug response. Suppose that can the tumor, can it Can the tumor, how aggressive is the tumor is? Can it quickly implant in the mice? Yes, the more quickly is it going to be engrafted in the mice, it could be an aggressive tumor. Same way, if the explant is it able to grow, very establish itself growth in the in the in vitro culture and respond to the drug, can be assessed, and this can be checked, and the endpoint analysis is to do for the different drug response assays, and also we can also predict the DNA, and the DNA changes and the RNA changes upon response to the drug. This slide is the patient-derived explants of glioblastoma, where we can clearly see that the patient-derived tumor, which has different cells, different other cells, and where at the same cell number of cells have been able to replicated, have been able to brought back to the explant culture. This is about with respect to the patient 1 and patient 2, where here we can clearly see that the difference in the different types of cells, where the heterogeneity, as we all aware, glioblastoma is a very heterogeneous tumor. So the different clonal distributions of cells is maintained in PDB cells, and the proportions can be clearly estimated. We will come back to this slide after we clearly understand what are the different preclinical models. The 2D cell lines, which we have clearly seen in our last session, and the different organon chips, the spheroid cultures, the organoid cultures, and the patient-derived monograft. Each one have their advantages and disadvantages, where the organoid chips can be grown in a controlled environment mimicking the host in vitro, host in vivo conditions, and it can be facilitated tissue-tissue interaction, and we can even have the ability to choose a co-culture where you can have have two cultures growing here. However, it could be very much it has to be, the conditions have to be very much well, they have to be could be very inconsistent, and mostly this particular model is used for cell lines, and sometimes it's very difficult to establish protocols for some tough models such as your lung. Then the spheroid cultures, yes, a zone of necrosis can be seen, and we have umpteen plasticware in the market to help us generate spheroids for any cell type. So though the protocols are simple, it has even a co-culture availability, and it can be reproduced at very high much because using the cell number. Suppose you have used only 2000 cells or 5000 cells, yes, it can be highly reproducible, and it can be patient specific, where we take up only the cells from a tumor cells and you generate, for example, a mammo smears. The poor structural organization and the poor supply of oxygen to the nutrient core and the to the spheroid core and the variable spheroid size and the variations in between patient to patient type could result have a very great disadvantage on taking this assay to high throughput levels. Again, we have the organoid co-cultures, where multiple cell lineages, complex structural organization, and patient, they are very patient specific, and which we will be seeing in the further on in this particular session. However, the same disadvantages as with the spheroids, mammospheres, highly interpatient variability there is, and it is very tough to bring in a downstream experimental analysis to scale up the downstream experimental analysis, and the high cost to make it is this organoid cultures, always to use it in a very economical setups. And then coming to the patient derived models, which we will discuss it in the next class. So this particular patient-derived explants, as the first human histoculture studies were first, as we have mentioned before, they were pioneered by Robert Hoffman, and they involved the generation of tissue slices from tumors obtained directly from surgery from the operation theatre, when they cultured for extended periods of time using the specialized collagen gel. So this was a very fantastic idea, and this ensured that the stromal component of this particular surgically resected tissues is maintained. So and it is maintained intact. So cells were found to maintain properties of their both in vivo state, which includes the spatial organization and the cell differentiation function, and the and the presence of more of say many cell types within a single tumor. They are all well retain. So histocultures, they were also called patient-derived explants, were also called earlier as histocultures, and they were adapted for use in drug studies to assess the chemotherapy response, and were successful. And this term was also used as the HDRA, that is your histoculture drug response assay, for assessing the chemotherapy response. So HDRA studies reported around 86% accuracy in predicting the drug resistance, especially for your colon cancer and the stomach cancer. Correlation for the drug resistance, the clinical trial studies to clinical drug sensitivity in all these two particular cancer was very well high. Several studies have employed this PDE cultures to lung cancer. So there have been, all that they had required is to require a gelatin sponge or a collagen sponge, or to use a matrix grid. But very well in the market, so many grids are available where this explants can be cultured using using this particular grid. And there is also another submersion method where where the tissue is completely submerged in media in tissue culture dishes, by the grid method, in which the tissue is in contact with media through the matrix membrane, or a membrane supported by a plastic or metal matrix grid, or by the sponge method. So even though several methods have been developed, this has been very well exploited to directly study the efficacy of the drugs on the tissue model for any tumor. These protocols are very simple, and they can be extrapolated for lung, oral, or for breast cancer. And they have an advantage, have been advantage because they can facilitate all these sponges, the collagen bases, they have an advantage because they will they have been as they facilitate the distribution of medium, nutrients, and drugs through the explants without having the explant to be submerged in the medium. They also may be maintained as fragments. The tissues can be continued to be maintained as fragments or processed for general tissue slicing. So this is all with the very well important, so different milestones which are given here. So we have already clearly discussed about the different particular advantage and the disadvantages of this particular model. So all that we have to be stressing here is that there has been, even all these clinical models or whatever the preclinical models are very important, because we will be seeing in the next session how so many compounds enter the trial phase here, but they fail to come to this particular, there's lot of iteration rate. So to reduce the iteration rate, or to check for the better efficiency of different efficient preclinical models have to be developed with less complexity, and to have a very scalable, high throughput procedures. And most importantly, all these models, preclinical models, have to be having a good lifespan, and they should be amendable for large tissue scale, tissue constructs generation, and for tissues that are they should be suitable for tissues that are physiologically at air liquid interface, such as the skin and the lung, where it's very important to maintain that ALI. And they should, and majority, they should be able to have a very high reproducibility. For example, the same tissues, they should be able, if you are able to propagate them for several generations, they should be able to give the same kind, each generation should be able to give the same kind of drug response to at each particular time. Here we will be checking about patient-derived explants of glioblastoma. Glioblastoma is one of the most lethal human cancers, and represents a solid tumor which is very heterogeneous in nature, and it is a glioblastoma multiforme. Although glioblastomas have been characterized at the molecular level, and several targeted therapies are there, they have highly little impact due to the heterogeneity and the plasticity because of these particular cancers. So, and for several generations, there have been only cell line models, or several decades, only cell line models have been employed for glioblastoma for the clinical models. However, one particular group, LeBlanc et al., they described a 3D culture method that used patient-derived explants and which allowed the retention of retention and recapitulation of the genetic and phenotypes of the original tumor. And they have gone about to, so they have, as mentioned, tumors contain heterogeneous neoplastic cells and diverse stromal elements, and that collectively function at the dynamic ecosystem, and this complicates the predictive modeling at the ex vivo level. So, this particular LeBlanc et al., they have used a single cell analysis to demonstrate that the patient derived explants replicate the tumor diversity as it can be represented here. Here we can see how the complete tumor diversity is retained in the patient derived explant, whereas the gliosphere cultures of the patient 1 and the patient 2, which are only getting only the small sample of the cells from the tumors. Glioma is one of the most lethal cancer, and glioblastoma represents the solid tumor with striking intratumoral heterogeneity, and it has been termed as glioblastoma multiforme. Although they have been glioblastomas have been extensively characterized at the molecular level and several targeted therapies are available, but for the tumor researchers for several decades, they have been using only cell line grown in serum or in plastic as models, because there is little and there is always been a little scope for developing this particular models in an ex vivo cultures, but there is little evidence that these cell lines have similar tumor genetics and transcriptional recurrence or in vivo tumor growth patterns to those of these cancers. So, in this particular group by LeBlanc et al., they describe the 3D culture method that uses the patient derived explants and of the glioblastoma, and this had allowed to retain the phenotypic heterogeneity as present in the tumors. So, there has been the continued presence of non-neoplastic cells from the patient tumor and the heterogeneous transcriptional cell state distribution, which are termed as the metastate. Tumors which contain heterogeneous neoplastic cells and diverse stromal elements that collectively function as dynamic ecosystems have been able to be replicated in this particular PDE models, and this particular, because of such existence of this, the neoplastic cells and the metastases together has been very difficult for researchers to establish the PDE models. So, the patient derived explants can be as good as tumor models. This particular group has been able to compare cellular fidelity by using the single cell genomic and the transcriptome sequencing to trace the populations present in the original tumors through their establishment of the PDEs and the subsequent PDE passages. Here we can see how the tumor cell metastasis, and if you can see only some of them are retained, so it may not be here uniform, whatever the whole tumor TME, the stroma elements along with the other cells, other non-malignant cells are not particularly integrated in the particular glioma spheres cultures, but in the patient derived explant cultures the whole tumor can be mimicked and it can be established as culture in the in vitro scenario in the lab, and the same targeted therapy the patient is taking can be administered here. This is a workflow for a typical PDE culture. Here we have a tissue processing where the directly surgically resected tissues which has to be brought from theatre, and then that has to be cut into small 1 mm or 2 mm bits, and it has to be recovered by giving washes in a Hank's balanced salt solution and washes with the particular antibiotics, and then it is established either in this particular metagrid or in particular collagen or in the dental sponges. However, And after they have been established for 24 hours, the cultures, they are going to, we will be treating them with the drugs, the different drugs such as whatever, for example, if it is a TKI inhibitor for lung cancer will be given here, or if I have to try any different combinations, they will also be administered here. After the required time, necessary incubation time for, say for example, which will be standardized 24 to 74 hours, the tissue sections will be fixed and will be embedded in slides, and then we will be labeling it for different markers such as I61 or the PrEP here. And here we can clearly see the control population, the drug, in the presence of the drug, how the different the PrEP and the Ki61. Here we can see that the absence of this particular marker. The application of the tumor mask and the digestion of the image, it can be it can allow the segregation of the staining in the tumor and the stroma. The graphs at the bottom, they depict the four quadrants, which shows the percentage proliferation and the cell death. That is your the Ki67 and the CPARP here at this particular quadrant. In the stroma tumor for the non-squamous cell lung carcinoma PDEs, which are treated with your vehicle control and with the drug. This is a typical workflow of this is a word for the complete PDE models, more complete protocols, and it is very important to have this particular marker Ki61, which will show the proliferative index. So, and then both the PDEs retain a expression of genes with the oncogenic roles in glioma, and they maintain the similar clustering patterns as with the same host tumor regions from the single patient samples, or also from the same different patients. Therefore, compared to the parental tissues, PDEs show consistent and potentially predictable changes in the gene expression of selected processes. This is the strategy to establish for the ovarian cell line. So for the ovarian cancer, for several all solid tumors, mostly the PDE can be easily established, where from the after the surgery restriction from the ovary, there are, as I mentioned before, with the using in this, all conditions have to be done in a sterile conditions, like as for the same cell culture requirement will also be enough for growing the PDE, for establishing the PDE models. After the tissues are minced to 1 to 0. To 0. mm squares, they are either grown in this orbital agitation, or even in, as mentioned before, in the sponges or in other metal grids. And then how are they characterized? After exposing them to the drug, they are so we have the control, which is untreated, the carboplatin and the paclitaxel, which are given to the patient for the administration during the patient, the same the PDE models also. Here you can get a picture. First thing, for the patient to see the outcome of the drug, it may take a very long time, whereas at this particular stretch here in the PDS, within 72 hours, if the whole tumor has been used in the right conditions. I mean, first thing we have to make in point, the tumor should be immediately placed from the patient, immediately it should be established in culture. Then only we can be able to correlate the drug response of the tumor, of the PDE model and the tumor. After we characterize them, they will be established for the proof of concept drug challenge. Now, coming to another very important preclinical model is the organoids. What are organoids? Are three-dimensional structures of the organs, and there are many organoids developed for all the solid tumor molecules, smaller tumors such as the brain, the lung, the stomach, the breast, and so on and so forth. These organoids have opened the new avenue for development of novel and more physiological human cancer models. Such clinical models, they are essential for more efficient translation of basic research into and also for bringing in novel treatment regimens for cancer therapy. So wild type organoids can generally be grown from embryonic and adult stem cells and these display self-organizing capacities and with phenocopies of the essential organs from which they are derived from. So genetic modifications of organoids has allowed for disease modeling in a setting that approaches the physiological environment. Or organoids can be grown with high efficiency from patients derived from healthy and tumor tissues, and they potentially enable, as mentioned before, lot of drug screenings and the development of individual treatment regimen. So, first thing, the Sato et al., they developed the 3D epithelial organoids and they have established from the LGR5 interstitial stem cells. So, and what they have essentially found out that upon embedment of the stem cells into matricels, cells can be cultured under serum-free conditions, which mimics the in vivo stem cells niche and which involves this particular WNT antagonist and the ligand of LGR5. This is very critical finding for the development of organoids. This culture protocol formed the starting point for other organoid cultures for multiple mouse and human epithelium, which includes the colon, liver, pancreas, prostate and stomach. The fallopian tubules, esophagus, the breast and the endometrium have got very well organized organoids developed. Organoids can be expanded long term and they are very well advantage with them is they can be cryopreserved, and as mentioned before they can be genetically modified, and these modified organoids are both genetically and phenotypically stable. Organoid banks are very well available in across many top labs of the across the world. This is the scheme of a stem cell driven tissue renewal in organs such as the intestine and stomach. As we all aware, a stem cell concomitantly self renews and regenerates, rapidly dividing into transient amplifying daughter cells via asymmetric cell division. This transient amplifying cells undergo several rounds of division before differentiating into the functional cell types of the adult tissue. Adult stem cells potentially can follow three modes of cell division: symmetric division to generate two stem cells, asymmetric generation to generate one stem cell and one TA cell, or a asymmetric generation to take the two TA cells. As mentioned before, organoids can be established from both patient-derived healthy and the tumor tissue cells. Here we can see the four types of organoids: the healthy organoid, the hepatocyte organoid, the tumor organoid, as represented by the different colors. For example, given drugs, both drug A, B, C are treated in this particular, all the three in the including your epitopic cell. It could be, and we can even check the toxicity of the specific the specificity of the targeted therapy. Is it specific to only the tumor and now not harming the healthy cells? And can be and in the same way, the liver organoids can be checked for the liver toxicity testing, which is very vital part for the drug pharmacokinetic studies. They can be used in genomics, the organoids. They can be we can study the complete genome of each organoids, and we can continue to have this living organoids biobank, which is very well prevalent. This is the role model for how a complete personalized medicine for one particular individual can be taken up forward for testing the efficacy of the different drugs, such as this particular, as it's shown here. So because it is toxicity to the drug A is toxic to the normal cells, drug B has been approved here for the choose the drug B for to treat the patient. Organoids can be generated both from the tissue derived cancer cells or the induced pluripotent stem cells. So to generate organoids from the TDCs, tissue samples are obtained from the humans or mostly the mouse, or also the mouse models. And the first, it is the interstitial tissue was the first organoid model to develop. So here the interstitial tissue will be opened up The tissue samples the interstitial tissue samples are opened, washed, and cut into small fragments at such as to 2 to 4 mm to increase the surface area for enzyme digestion or further mechanistic processes. This particular digestion will allow for the isolation of single cells or single interstitial stem cells or crypts. After several rounds of washing and purification, the harvested stem cells or the crypts will be used for generating the organoid cultures for expansion. And this is further can be further processed by addition of several particular growth medium and differentiation factors, this particular organoids can be generated. To generate organoids from the induced pluripotent stem cells, genetic engineering can be used. So here the iPSCs are maintained and expanded as undifferentiated clonal populations on particular feeder cells, or particular extracellular matrix substrates to aggregate to embryoid bodies. Typically, embryoid bodies are formed. So, aggregation to embryo, as we can see here. So, typically, induced pluripotent stem cells are harvested as cell aggregates which preserve the cell-cell contact and yield cell populations with very high variability. These aggregates are further induced, as we can see here, through germ layer specification to form endodermal spheres, which can be seen here. Endodermal spheres, then the mesodermal spheres, and the neuroectodermal matrix for additional expansions. The starting population for organoid cultures are generally obtained from the adult tissue or adult or fetal tissue biopsy samples. This is very important process for the procedures for development of establishment of organoid cultures. And coming to the different organoid types, the tissue fragments and the explants are generated from the original cell types and ECMs, where the physical connections and the paracrine signaling networks are maintained. This is very important for if a tumor tissue fragment or a normal healthy tissue fragment is taken. It is a challenge to isolate to ascertain the contribution of an individual cell type. It's definitely a challenge to establish in the progression, in the growth or the response of the organoid to that particular drug. And this particular tissue fragment organoids display high patient to patient variability, whereas the reconstituted organoids are assembled in vitro from cultured or sorted cells. This permits expansion and modular assembly of components. However, some subpopulations or phenotypes such as senescent and hormone responsive cells may be lost during culture. So, it is very important to maintain the particular growth factors along with during this culture, and again it may be variable or it may not be very much uniform across all organoid formations. And another type, stem cell derived organoids, are differentiated from the multipotent adult tissue stem cells or the PSCs, which we have seen under different conditions, cell culture conditions. PSCs specifically can generate rare tissues and cell types, but have limited maturity and they lose the hallmarks of aging. They have reliance on exogenous growth functions and differentiation protocols. These are the different types of organoids. If you recollect our first slide, where we have said that the first organoids are to be developed are the for the interstitium. So the first this is a typical protocol for patient-derived organoid cultures for human colonic mucosa, where the mucosa is fresh human mucosa, which can be where the epithelial crypts are and they are allowed to establish as organoids. And here we can see after growing for some time in the matrix, we can see the epithelial cell, the central lumen, and which this is all embedded in the matrogen. Thus, they as critical player is the in the establishment of organoids is the stem cell, which is a self renewing cell, and it can give rise to different cell types within the same tissues. So they have, as I mentioned in the first class, they have the unique property of having a leucine rich repeat which contains G protein coupled receptor 5. So once organoids have been established as a different, as mentioned before, they require complex and individual combination of growth factors, survival, for the survival and maintenance. Some of these growth factors are also obtained from the patient's autologous serum or from the same body of the body fluids from the originating tumor patient. So, organoids for different tumors will have different growth requirements. Subtle changes to these growth requirements have to be modified from based on from lab to lab. For example, normal organoids will outcompete colonic cancer organoids when cultured medium if it is used for normal colonic organoids. So it has to be potentially separate from which has to be different for cancer colonic organoids development, because there is, however, the sensitivity to of the organoids to the growth factors. The sensitivity of organoids to growth factors is very vital, and it can be used to employ many of the normal colonic organs. For example, the normal colonic organoids, they require the ligand the WNT3A for survival, whereas whereas as we are aware, the colonic cancer cells, they demonstrate hyperactivation of the Wnt beta catenin pathway, which is independent of the WNT Wnt3A. Therefore, selective removal of the WNT from organoid medium prevents normal colonic organoids from outcompeting the colonic cancer organoids. So, not all colonic tumor cells, they display aberrant Wnt signaling though, and therefore it is important to exploit or to explore the implications of selecting tumor cells by the requirements for specific factors, as demonstrated here, the Wnt factor for colonic cancer. So it is very important to use different to use growth factor requirements, and to be informed of the growth factor requirements to characteristic, rather than also the selective rather than the select human tumor organoids. To alter the organoid functions, the growth factors selection is important, and also the altering the concentration of that particular growth factor in the medium is important, and the two important to characterize the organoid cultures before experimentation. So as we can see here, how the growth enriched medium embedded in extracellular matrix, and it is, you have the patient-derived tumor organoids formation here. And once, what is the state endpoint analysis? Can we check for the correlation from the original tumor before the organoid is formed? It will be correlated with the original tumor, and then if, for example, you will characterize the whole genome sequencing or the exome sequencing or the proteomic profiles of the whole tumor and match it with the organoid form. And then again, this particular organoids they can be they can go through the process of cryopreservation and also through paraffin embedded molecule. So several research molecules for research protocols can be streamlined using this particular organoids for one particular cancer molecule. This slide shows the downstream processing of the organoids for further characterization. Here, as I have mentioned here, maybe for this is the liver organoids. Here we have the healthy liver and the different liver, the cancer liver, all that they have been taken, they've been processed in the lab for establishment of organoid cultures. So isolation of cells from patient samples and organoid cultures are all happened here, and further even the 3D culture or expansion also can can take in place parallelly, and the organoids are biological and molecular characterization Here is the typical characterization of cancer organoids from liver, where the normal tissue and the chronic cancer tissues, they're all collected, established as organoids, and the DNA after the dissection and the RNA after the dissection, the establishment of the normal healthy cancer tissue, as well as the cancer healthy tissue, as well as the tumor cells. The organoids are established, expanded, and the DNA and the RNA and the histology is taken up, the DNA and RNA for the whole genome sequencing or the transcriptome sequencing respectively, and the histology for different pathological markers is taken. Here this particular slide gives the histography for the HND for the organoid and from the tissue, where a lot of characteristics from the for especially for the cancer tissues, they are all retained both in the organoid as well as in the cancer tissues. So the histological analysis of this liver particular analysis, the top row shows organoid bright field scales, and the bottom row shows the HND staining of organoids. So here this shows the analysis of specific marker expression, especially for your AFP, that is the hepatocellular carcinoma, where the HCC stands for, and the EPCAM, that is the for the CC marker, and the DAPI staining, which shows in blue. So organoid or propagation is represented as growth and splitting curves, where dots represent the splitting time point, and arrows represent the expansion here, which we can clearly see how the healthy one and the LT2 we can be clearly mentioned here. And the analysis of the genetic changes can be clearly seen here, whether if where the light blue measures shows that this particular tissue is in concordance, the DNA analysis shows that the 80% of the DNA is in concordant with the original tumor, whereas this particular is the one in red, which is a very small portion, which we can clear as typical organoid specific, and with some of them is only the tissue specific. So by and large, majority of the characteristics of the original tumor are retained in the organoids, and even the drug resistance also for the particular concentration can be seen. For example, gemcitabine here, which is used here, and for the different conditions, the organoids and the responses can be clearly shown here. So transplantation into, suppose this, if these organoids can also be transplanted into the mice models, which has shown that and they get the histology and the xenograft analysis matches the patient's original tumor tissue. Here the pancreatic island organoids is represented here. As I mentioned before, these organoids are very well growth specific for the growth medium and the growth factors they are growing in. The organoids growing in the matrigel, they exhibit here the spherical shape as shown here, and these spherical organoids are surrounded by an endothelial cells, as we can and as we can see in the higher magnification here, which is represented here. And this expression during the microscopy, we can see immunofluorescence or histochemical staining, the expression of insulin in insulin beta cells, glucagon, that is your CCG alpha cells, stromatin, and pancreatic polypeptide can be clearly seen in all this particular matured organoids. The morphology of the mature organoids induced through prolonged culture for a total of 30 days here in this particular experiment. The organoids are surrounded by the endothelial cells as shown here, and they are shown by the CD31, the endothelial marker, as we can clearly see here. Staining for some of the key transcription factors and differentiation markers is also shown here. There have been for 3 rounds. This shows the protocol, how the organoids are maintained, where the pancreatic island organoids, they function, their functionality is validated vitro in vitro, where organoids are subsequently sequentially incubated with low and high concentration of glucose for 3 rounds, and they are washed, followed by C peptide ELISA and calcium imaging here. This rise of the C peptide here is here, as we can clearly see, is indicating of a sharp response to offers glucose stimulation. The different, and here this G represents the representative curve of intensity of the calcium signaling, indicating the organoid's capability of responding very well to glucose. G represents here representative curve of intensity of calcium signaling trace imaging, which indicates that organoids are capable of responding to the glucose with high or low calcium activity, which is consistent of the high or the low glucose stimulation. So very well illustrated pancreatic island organoids, which has been reported. Finally, this what is the endpoint analysis after this particular? This organoids can be can be gone for many generations, as mentioned before. They can be even be given in taken. This organoids can be taken with the organoid transplant can be done, and and first thing, they can be given into the mice models. This is the very important. This organoids or fresh islets as positive control are transplanted into the kidneys of a capsule of streptozotocin induced diabetic mouse models. Islet organoid functional validation, here it is happening in this mice models. So organoids or fresh islets of pancreatic fresh islets as positive control, they are transplanted into the kidney capsule here, into the kidney capsule of streptozotocin induced diabetic mouse, followed by monitoring of certain physiological indexes which indicates the rescue of the diabetic phenotypes. Finally, these transplants are removed and the surging blood glucose level in the mice are noted by the plasma insulin weight and body weight. So here, so organoids are so we have started here with an in vitro here, and then moved on to an in vivo conditions here. This is how the pancreatic islet organoids are well established and used in diabetic mouse models. A recently developed method called conditional reprogramming. It facilitates the establishment of 2D cultures from normal and tumor epithelial cells with very high efficiency. These cultures can be maintained long term, and they are known to retain a stable karyotype. There is a presence of there is a need for the presence of an ROH kinase inhibitor and the fibroblast feeder cells. So patient derived tumor xenografts, they have the advantage of as we are all aware, have the advantage of mimicking the biological characteristics of human tumor much better than the organoids model. The ability to serially transplant into to ability to increasingly so can, as much as we can passage many organoids, can be also passaged in mice model molecules. So they have been this organoids, or the whole purpose of this slide is to emphasize that the patient derived explants or the patient derived organoids, that is or the patient adult stem cell derived organoids or the tumor derived organoids are very important in to first establish a preclinical model, and then to move a mouse model, as mouse models is more costly, requires a better skill set, and it involves the use of limited animals and limited engraftment efficiencies for subset of patient tumors. So, and moreover this so it's always, so here we can clearly see whether there is a conditional reprogramming for this particular cells programming for this particular cell lines or the particular patient divided organized the PDX. We have discussed all the several advantages and disadvantages of using the organoids or the PDTX, but here we would clearly emphasize here only on this particular point. It is that they are expanded, easy to expansion is much well achieved only in the cancer cell line than as compared to the other models, and the 3D growth patterns are also very easy for the cancer cell line as compared to the other models. And only this PDX and the patient-derived tumor models, they retain the original tumor interactions, they incorporate the immune system, and they have the genetic cancer modeling. And but however, they lack even some of this particular PDX model, they lack the high throughput drug screens. So tumor organoids can be manipulated to improve the functional utility in cancer research. As I have mentioned before, they can be genetically edited through the CRISPR-Cas9 technologies. They can be genetically through the CRISPR, they can be established in co-culture with other cell lines, such as your tumor, that is the immune cells. Then you have the endothelial cells, stromal cells, and they can be micro injected with any other growth microbes or antigens or chemicals. Then some several experiments can be used to reveal novel insights into cancer pathophysiologically, as we have shown in the before. And then immunohistochemistry can be easily done on this particular organoids. And then even you can even bring it up, suppose you have the single cells, you can even bring it up in a co-culture where you have the insert. Maybe you can have another cancer cell line or another organoid here, and then you can establish it in co-culture, and then they can be done easily flexible for immunofluorescence, as I have shown before. And then they're very useful scope for organoid on chip, which we will be discussing later, and establishing of the xenografts. So very quickly we will be coming to, because it's a very vital part, and which is how the whole direction of the preclinical model, developing cancer preclinical model is moving to. We will be talking of the single organ on chips or the multiple organ on chips. So first, as I mentioned before, gut on a chip device for drug absorption studies was established before, where here a transfer cell culture cell culture insert, that was the inspiration for this gut on a chip flowing microsystem. Here the drug in the medium, it will be just drug in the medium. Here the drug in the medium will be in the apical side, and whereas the medium will be on the transport to the basolateral part of the slide of the dish. This is the co-culture insert. And then intestinal absorption of orally administered drug is commonly evaluated in this particular transfer assays, where a layer of interstitial epithelial cells is cultured on the permeable here insert placed in one of the wells of a cell culture plate, as you can clearly see here. Then the intestinal cells act as a barrier between the top the intestinal cells is cultured on the permeable in one of the wells of the culture plate. These intestinal cells act as a barrier between the top compartment, mimicking the gut lumen, and the bottom compartment, mimicking the bloodstream. Drug absorption can be measured over time by analyzing samples taken from both the compartments. This has been intestine, you suppose, imagine in this co-culture, this requires a typical 6-well plate or a 35 dish, and if this can be taken up into the microfluidic channel level, it can be taken up, it can be easy for handling, it can be easy for large scale drug assays or high throughput screening. These interstitial cells act as a barrier between the top compartment, mimicking the gut lumen, and the bottom compartment, which mimics the bloodstream. And drug absorption, as mentioned, can be measured over time by analyzing samples taken from both compartments. This has been translated into a microfluidic format, as I mentioned before, where the interstitial, where the barrier consists of a PDMS membrane with transwell with a microchannel containing the dynamic flow of cell medium, and which is, this is still in conceptual and design. The PDMS is used because it is here, this particular membrane, it is used because it is transparent, biocompatible, and and it is a moldable silicon rubber which molds and these molds are produced either by the photolithography or by 3D printing. And so it is here, if you can clearly see how the engineering technology has come into aid to develop this organ on chips devices. Here the cathode cells, the golden standard cell line for drug absorption studies, can be cultured here in this particular we can clearly see here, can be cultured here, drug absorption studies. And these cells may lack many other functions that interstitial epithelial cells have, but they are very suitable for absorption studies. The interstitial organoid chips are kept in the incubator with the cell medium provided by the 2 syringe pumps without the recirculation. This device can be inspected by optical microscopy, and the permeability of model drugs will be monitored through high performance liquid chromatography and fluorescence based assays with the samples of organ on chips format, including the testing for even your antiviral drugs have been very well developed using this particular models. So, for example, the process of viral infection is far more complex and requires multiple different organ functions, including immune components, to be present. So, a similar 2D organ on chip cancer device was used based on the porous membrane, but with the inclusion of more biological functionalities such as the function of both the interstitial epithelium and the blood vessel wall and the immune component have been used. Here this B represents the multi-organ on chip models, which includes 4, 7 or 10 organ models that can interact one another via the microchannels, as we can clearly see here. Here you have the gut, the brain, the liver scaffold, and this is all very well engineered. The different various layers, there are different connections for the cell culture medium and pneumatic channels to acute integrated microparts. The different approaches for multi-organ organ on chips for drug metabolisms was developed, and it is aimed at developing a physiologically relevant multi-organ system for drug discovery. This is very vital. The system embedded 4, 7 or 10 organ models, as we can clearly see in here. This integrated device was fabricated with easier to process PS, that is your polysulfonate, and polyurethane with integrated air pressure control valves as built-in pumps to recirculate medium in a more controllable fashion. This a lot of materials engineering and lot of 3D printing science has to be involved. So in this process, the organs models were of human origins, primary cells from cancer from cell lines or from IPSCs, immunopluritin stem derived cells, and included the kidney, muscle, liver, and gut, among other organs here. The pumping system was incorporated into the final device, as we can clearly see here. The complexity of this particular system has made it necessary to answer a more complex scientific question. But it is like you need to have so much of engineering in technology, but the various pieces of so much of supporting equipment which is required to maintain this particular organoids in an active state or in live state, and there is a lot of additional cost for setting up this particular system. Then they came up with a miniature system such as these, for a miniature for testing for toxicity testing of various anti-cancer. This system contained a reconfigurable design with multiple organs and includes a solid tumor derived cells and also leukemia to study the effects of anti-cancer drugs on their target, as well as the the study of the anti-cancer drugs on for the toxicity of this particular cells of the compounds on other organisms. It's still in the conceptualization and the design phase. And cancer cells modeling various malignant conditions are cultured in all this particular organ on chips similar to other systems. This device is more or less pumpless and hence more user friendly, and it doesn't require more supporting machines as this particular platforms, and it can be placed in a rocking shaker for a gravity induced recirculating flows, or and therefore they're reducing lot of the laboratory technical machine which is required for this maintaining this of particular platform. So multi organoid on chips are very developed, consisting of several organoid on chips compartments together in one device, and this is composed of microfluids, microchannels and chambers with even you even have the green dyes also to be filled to for visualization and observations. Quickly we will be going through here, even though you have the different how do we collect the biological results from organoids on systems. As I had mentioned before, there is an integrated sensors, and there is a Maya. This is a very famous term for organoid on chips here, the microscopy and the high content imaging. It is very important to collect the biological material from the organoids of organ on chip systems from time to time during the during the cultivation of organoids. There are several in situ measurements during operation of the recirculating organ on chip systems, and these allow near real-time assessment of cell states and functionalities. The integrated electrodes and the cantilevers in the organoid on chips devices, they can be configured to measure the electrochemical changes or the signals which are indicative of the transepithelial electrical resistance, as we had mentioned here, which is very important for to using the microelectrode arrays and to measure the cell tissue contractility using the cantilevers as in the AA as measured here. There is high microscopy and high content imaging are also among the most widespread analytical methods used for and they find a very they can be in very good visual inspection for organoid on chips. They are all this organoid on chips, they are all usually constructed with glass materials or fiber materials that they are easy to be held on stage in the microscope, and they are where they are micro optic friendly, and they will be falling within the imaging depth of the conventional fluorescence or the confocal microscopes. It is very well convenient to perform in situ staining or of the cells and tissues in chip with fluorescent stains or antibodies to assess the cell viability or expression of specific tissue specific or cancer specific biomarkers for this organ on chips. And all this particular microchannel fluidics allows for the delivery of this particular reagents and dyes into the to the particular organs. High content imaging, everything is possible for this particular organ on chips. Yes, as I mentioned before, different analytes using LCMS and other technologies, they can be employed here. We can even use the downscreening for your whole genome sequencing, the NGS, the RT-PCR for selection of different markers for assessing the organoid on chip. These are the current limitations of organoid on chips, where we have the accumulation of dead cells and debris inside the organoid lumens, and then there is limited control over heterogeneity or non-screenable or less reproducible morphology, which may not be relevant in the particular scenarios, or organoids do not recapitulate the cellular and physiological complexity of the native tissues. So, but there are definitely we can have still for example, for accumulation of dead cells and cell debris inside cystic in this particular cystic organoid lumina, they have been overcome by designing a perfusible open-ended structure that can use inducible flow to wash out. A flow is used here to wash out the dead cell debris, which are more compatible for long-term experiments. Organoids grown in matrigel, even though they display high variability of cell heterogeneity, as we can clearly see, and differences in morphology, but these platforms are, they can be overcome by using a grid here, as we can clearly see here, with pattern synthetic ECM that provide cues for. So you have a different, you have a specific UVS which can provide cues for cell differentiation. These platforms are additionally cell type derived. These platforms are compatible with high throughput screenings. Organoids do not replicate cellular and physiological complexity of the native tissues. They can be, technology would be enabled to have a controlled microenvironment, as it's shown here. For example, soluble 4 factors, or as I mentioned before, like here, WNT for the deprivation, for the for selection of your cancer cell, colon cancer cells over the normal colon cancer cells. And then even you have the oxygen also as one more very easy factor to regulate in the organ on chips. The organoids can be very well manipulated to require to suit the, to mimic the tumor microenvironment as per the host. We have come to the end of this session, and where we have discussed in detail about the different patient derived clinical models, which is, and both the patient derived explants and patient derived organoids. So two very important clinical models which can play a very good role for in the drug discovery or during the therapy of to predict the therapeutic outcome in the for the patient are very well discussed. We have discussed the limitations of both the PDEs as well as the patient derived organoids. In the next session, we will discuss the animal models, the mouse models. Thank you.